Welcome again. Today we will be discussing about the activation of the B lymphocyte. Initially we have discussed about the activation of the T lymphocyte, and then we are moving forward to the activation of the B lymphocyte. So as a mature naive B cell leaves the bone marrow following the successful rearrangement of their membrane immunoglobulin receptor, they recirculate throughout the body, attracted to the follicular areas of the lymph node and the spleen. So they are found normally in the follicular area of the lymph node and spleen. If antigen entering the secondary lymph organ, that is the lymph node and spleen, binds to and cross-link the isotypes of the immunoglobulin, this provides the first signal for the activation of the B lymphocyte. So you have to understand that B lymphocyte is actually activated in the two forms. One, B cell is itself a antigen presenting cell. So since it is itself antigen presenting cell, it can directly bind to the antigen. And if it is protein not in nature, so like lipids, carbohydrate, polysaccharide in nature, it will be directly activated, converted into the plasma cell and then form the antibody, but the immunity will be lower or weaker, and then there will be no memory cell. This is in case of Thymus independent, that is not in protein in nature. That will be directly activate the B cell and then form the plasma cell and then form the antibody, and there will be no memory, that will be weaker immune response, and that will be again clear out, that will be resolved, that will be damaged or diminished from the body soon. But if it is the Thymus dependent and protein in nature antigen, what will happen? That will process and presented by the MSC class 2 molecule, that is anti-presenting cell, that will reach to the T cell, and then T cell will again come to the B cell and then activate the B cell. So in that situation, the B cell will convert into the plasma cell. Since they are a protein nature, that has been activated by the T cell. So T cell once activate the B cell, then that will convert into the plasma cell as well as the memory cell. So there will be two things. There will be the plasma cell and memory cell, and since there will be a plasma and memory cell, they can form the antibody will remain longer period. If there is a second time infection or second attack, then the already memory cell of B cell is there, and that will directly convert into the plasma cell and form the antibody, and the robust immune response will be there. So if it is a Thymus dependent, the immunity will be robust, there will be memory cell. But if it is in Thymus independent, then there will be no memory cell. What will decide it is a Thymus dependent or Thymus independent? The nature of the antigen. If it is lipid, carbohydrate, polysaccharides, that will be direct, that will be Thymus independent. If it is protein in nature, then that is the Thymus dependent. So this should be clear. Thymus independent, they will directly activate the B cell, convert it into the plasma cell, form antibody, but weaker immune response, no memory cell. Thymus dependent, this will process by MSC class 2 molecule, that is on the B cell, a macrophage and antigen presenting cell, that is the dendritic cell, and this will come and process and present it to the T cell. T cell will be activated, then this T cell will again secrete the cytokine and activate the B cell, and B cell will convert it into the plasma cell as well as the memory cell. So if the B cell is activated by the T cell, then that will form the memory cell and plasma cell, and in this way now there will be the strong immunity, robust immunity in case of Thymus dependent antigen. Okay, now the antigen that binds to the B lymphocyte encounters are divided into two categories, Thymus independent and Thymus dependent. The Thymus independent is certainly a certain mature B lymphocyte are capable of being activated by the macromolecules such as the lipid, polysaccharide and lipopolysaccharide. So you have to be clear, this is the lipid, polysaccharide and lipopolysaccharide without having interaction, without having to interact with the helper T cell. So there is no involvement of the T cell, and this is Thymus independent, that is T cell independent. So this antigen are called Thymus independent or T cell independent antigen. They directly stimulate the B cell, proliferate and differentiate into the plasma cell. So there will be the antibody formation. This response of the Thymus independent antigen is generally weaker than response to the Thymus dependent one. So obviously, since this is there is no involvement of the T cell, so there will be the weaker immune response, and the primary secretion is IgM antibody and the absence of immunological memory. So this should be clear. Thymus independent, that is the lipid, polysaccharide and lipopolysaccharide. There is no protein involved. there will be the no memory cell, and there will be the generally weaker because no T cell has been involved. The thymus independent antigen may also act as the B cell mitogen, directly causing mitosis regardless of the cell antigen specificity. B lymphocyte activated by the Ti antigen are found in the spleen and the mucosa. So in the case of the spleen, in the blood you can find this all component, that lipid, polysaccharide and lipopolysaccharide, and since they are found easily, there will be the more stimulation of the B cell directly in case of the blood-borne pathogen, and that will mainly found in the spleen and some of the found in the mucosa. So the marginal zone B cell are found in the periphery of the splenic white pulp, and B1 cell associated with the mucosa and peritoneum. So they are found in the splenic white pulp in case of the spleen, and in the outside the mucosa and peritoneum they are associated with the B1 cell. So we have understood this thymus independent antigen, that is the no memory, not protein, that is lipid, polysaccharide and lipopolysaccharide, they have no involvement of the T cell, so there is generally weaker immune response. If you talk about the thymus dependent one, what is the thymus dependent? Mostly antigen introduced in the body fall into the category, and that is the thymus dependent. That is the thymus dependent antigen response to the molecule requires the direct contact of the B cell with the helper T cell and are influenced by the cytokine secreted by this T cell. So there is the direct activation of the activation of the B cell by the help of the T cell, that is the helper T cell. After cross-linking of the receptor on the B cell surface with the antigen, the material is endocyte processed via the exogenous pathway to generate the MSC class II peptide complex, and which is inserted into the membrane of the professional antigen presenting cell. So look, there can be a confusion, like sometime what happen, the B cell itself is antigen presenting cell, so they will process the antigen and then they will present to the MSC class II and then go to the T cell, then T cell will be activated, and again the activated T cell will be activated back to that B cell and convert into the plasma cell. So if it is the dendritic cell and macrophage, what will come? They will process the antigen, then bring to the T cell, and then T cell get activated, selected, activated, proliferated, and then they will activate the B cell, clonal selection and clonal expansion, and then plasma cell converted and antibody form. But if you imagine, it is already, if suppose the antigen is not presented by the dendritic cell or macrophage, but presented by the B cell only, then what will happen? B cell is antigen presenting cell as well. So they will present to the T cell will be activated, and once the T cell is activated, they will again activate back to the B cell, and in this way plasma cell is formed. So here they are talking about the B cell is itself an antigen presenting cell, so they are presenting that antigen to the T cell get activating, and then again back they are activating the B cell and converted into the plasma cell and forming the antibody. So simultaneously expression of the B1 is upregulated on the B lymphocyte, making the cell effectively present to the CD4 T cell in the area. Once CD4 T cell recognizes a specific peptide displays on the MSC class II molecule, then two cell form a conjugate. So this is the way, initially also there was, if the dendritic cell and macrophage come, then there was the first they recognize through the MSC class II protein antigen by the TCR, T cell receptor, and then there is a co-stimulatory signal, that is B7 and CD28. So here also B7 and CD28 is required because we know T cell is activated by the two signals, not a single signal. So to activate the T cell, by the antigen that is been processed and presented by the B cell will be, first B cell will bring the antigen in the MSC class II protein that will recognize the TCR, then the B7 will be upgraded and form the co-stimulatory signal with the CD8. Now T cell got activated. Since T cell is activated, they will again activate back to the B cell to form into the plasma cell by the CD40 ligand, and now then plasma cell is formed. So we'll discuss about that. Okay. So talking about the CD4 T cell is activated and differentiated into helper T cell. The helper T cell arranges the Golgi apparatus towards the junction of the B cell, leading to the directional release of the cytokine. Expression of the CD40 and CD40L, that is CD40 ligand, CD40L on the surface of the helper T cell is upregulated and then interact with the CD40 and the B cell to provide the second signal for B cell activation. Now they are activating back to the B cell. B cell respond by proliferating and differentiating into the plasma cell and the memory cell. This plasma cell will secreting the many tons of this immunoglobulin M into the environment, and that will be now responsible. The antibody is formed in the environment and that will be fighting your infection. So you can easily see over here, this is the lymphocyte, this is acting as the antigen presenting cell that will present this antigen. They are presenting this antigen. So this is the B lymphocyte. You don't think about the plasma cell now. You first think this is the antigen presenting cell as well. So antigen presenting cell, this antigen presenting cell presenting this antigen to the T cell receptor, that is the naive T cell. Now what happened? This T cell need to be activated. For that there is a co-stimulatory signal between B7 and CD28. After this activated, now T cell got activated. Once T cell is activated, what will happen? This will release the cytokines and then back activate the B cell. If it is the macrophage or dendritic cell, then this T cell will be activated and it will go in near the B cell and then activate. Since this B cell is itself presenting as antigen presenting cell, it will activate first the T cell. Once by the primary signal and secondary signal, if both signal are okay, the T cell is activated, then this will again send back signal by the upgradation of CD40 ligand and CD40, by which there will be the B cell activation. This is the second signal has been occurring. Now this B cell is activated. Since this B cell is activated, now there will be formation of, they will convert it into the plasma cell, and this plasma cell will now be activated B cell, that is the proliferation of the B cell into the plasma cell. Now this antibody will release, and now since this is released into the circulation and go to the infectious site, and then there will be fight against the infection and your antibody. So hope you have understood the activation of the B cell. B cell is activated. It can be activated directly into the plasma cell if it is processed or presented by the antigen presenting cell like dendritic and macrophage. But if it is acting itself, then first it will present this to the T cell, then there will be the co-stimulatory signal, primary signal, secondary signal. The T cell will be activated. They will release the cytokine that will activate this B cell. B cell will convert into the plasma cell and antibody will be released. So we hope you have understood about the T cell activation, this B cell activation, so which is very much important. So we are talked about B cell activation. Now let's move towards the few points like B lymphocyte activated through the thymus dependent antigen released in the two subsequent waves, that is released in the they are released in the two phase, you can say. The primary wave of activated B lymphocyte is comprised of a strictly IgM secreting plasma cell which leave the secondary lymphoid tissue shortly after being activated. So since there is a T cell activated, the B cell converted into the plasma cell, that is the thymus dependent. So they will release the IgM antibody, secrete the pool of plasma cell that will release the IgM antibody into the environment, into the circulation. So this is the one wave of releasing the plasma cell. There is another second way of activated B lymphocyte remain within the follicles of the secondary lymphoid tissue undergoing clonal expansion producing the germinal center. During the expansion, the clones will undergo affinity maturation and isotype switching. So what is happening now? Once you have released this immunoglobulin M in the environment, now this will remain for the short time. You need for the class switching as well. So you need hemoglobin G, you need immunoglobulin A and other immunoglobulins. So what happened? That second wave, that second phase of activated B lymphocyte are multiplying inside this germinal center, inside the you can say lymph node, and they are multiplying over there. And what will happen after this will be over? They will release the secondary, that's plasma cell, or secondary this activated B lymphocyte, that after affinity maturation and isotype switching. So there now you will release after a few days, there will be immunoglobulin G, immunoglobulin A, that depend upon the signal of type of infection and signal that has been sent. So that will depend, the second phase of B lymphocyte will be released by after affinity maturation and isotype switching. Now we have to understand what is affinity maturation and isotype switching. The term itself is saying that affinity maturation means it is the ability of binding to the antigen. So if the antibody have increased affinity to bind to antigen is called affinity maturation, and isotype switching means it is switched from immunoglobulin M to immunoglobulin G or E or A depending upon the signal. Okay, so let's go through the affinity maturation. So during the activation of B lymphocyte by the helper T cell, intense proliferation of B cell result in the formation of germinal center in the follicles of the secondary lymphoid tissue. These clones of proliferating antigen specific cell during the intense proliferation response to the B cell, random mutation in the coding of the variable domain maker, this is called somatic hypermaturation, and a single point mutation in the antibody idiotype. So this is a simple thing. There is a mutation in the idiotype. Idiotype is the binding portion of to the antigen. So if there is something mutation, this will have increased affinity to the antigen, so they can bind more tightly through the antigen. Since their affinity has been increased, then the cell expressing, so if the slightly altered idiotypes have increased affinity for the antigen, then the cell expressing will be selectively advantage in comparing to the binding antigen. So there is, after somatic affinity maturation or somatic hypermaturation, there is increased affinity towards the binding to the antigen by the antibody that will be released by this cell, or say activated B lymphocyte or plasma cell. Because this binding antigen serve as the first signal for proliferation, over time the clones of the cell with higher receptor affinity will begin to predominate in the germinal center. This clonal selection results in the predominance of clone capable of producing antibody with increased affinity to the antigen, a process known as the affinity maturation. And this means that although isotype switching will necessarily decrease the ability of the predominance of antimolecule as the immune response evolves, this will substitute by increasing antibody affinity over time. There is another concept as well. Since you know immunoglobulin M is a pentavalent, it can bind to the many antigen, so it has more avidity. So avidity means binding, total antigen binding capacity. So obviously immunoglobulin M has more in comparison to the immunoglobulin G and E, because immunoglobulin M is pentavalent. But once class switching occurs, this will convert into immunoglobulin G and A that has less avidity. The total binding capacity decreases, but since it has increased the ability to bind the antigen, the affinity has increased, this will actually compensate. So this will substitute by increasing the antibody affinity over time. So increase affinity over the antigen, this antibody affinity has increased towards the antigen, so that will balance your avidity, that will balance the pentavalent which has more binding capacity by the immunoglobulin A or immunoglobulin G. Isotype switching, you know, this is one of the way by which one type, suppose immunoglobulin M is released initially, that will switch to immunoglobulin G, E or A depending upon the signal. Although all of the antibody molecules secreted by the clone of B lymphocyte have an identical idiotype, B cell is induced to make new class isotype of immunoglobulin in response to the cytokine directed instruction from the helper T cell. So initially by any reaction, any activation of a B cell will release the immunoglobulin M only. In acute infection, there will be immunoglobulin M only, but the isotype switch will occur by the cytokine directed instruction. So there will be a cytokine directed instruction from the helper T cell which leads to the isotype switching of the immunoglobulin. As the B lymphocyte receives cytokine signals from the helper T cell in secondary lymphoid organ, it is induced under the isotype switching, changing in the heavy chain constant domains to class of the antibody with a new and effector function. So what will happen? There will be the isotype switching will occur in the heavy chain, you know, and that's it does by rearranging the DNA encoding the constant region of the heavy chain by activating switch region that causes the intervening DNA to be loop out, excised and degraded. So the idiotype is then joined. You can see over here. This is your initially VDJ combination. First is the mu, so there will be the immunoglobulin M is released. After that, the D that will lead to immunoglobulin D, gamma that will lead to immunoglobulin G, then alpha is immunoglobulin A, and epsilon is immunoglobulin E. So it depends upon the signal and the looping out. So since this was initially from mu,